Welcome to the CSEC Drawing Lab from the cover page. We want to look at drawing as a skill for the SBA component. It is indeed a very important aspect of your SBA as it contributes 10 marks out of the total 70. Now, ORR would be contributing another 20, PD would be contributing 20, MM also would be contributing 20, all of which will be scaled to 30 marks. Then there's AI, which also contributes 20 marks, and that too is scaled to 10, which makes that total of 40 marks that you can earn for your SBA activity. Now here we have a little calculation to show you how we calculate that total SBA grade. Now you might wonder how many labs you'll be doing for the SBA. A minimum of 18 should be done, and the amount to be submitted for CXC, it's about 8 depending on your teacher. There are some labs that must be covered in the lab book to be submitted: ecology, movement of molecule, photosynthesis or respiration, food test, germination, disease and nutrition, and genetics. All these must be a part of the set of labs that will be submitted to CXC. Now, requirements for drawing. If you are going to draw, there are some basic stuff that you need to have. You need to have white paper. You need to have a HB pencil or a mechanical pencil with fine point that can give you a neat or clear drawing. You need to have an eraser should there be any error. You need to have string. If you are measuring something that is curled, then the string would come in very handy to form that curve and give you that accurate measurement so you can do your magnification calculation. It would be wise if you have an unlens. So if you're drawing very small things, the unlens would come in very handy. You need to have a ruler. If you are drawing non-organic shape, a ruler would be very nice, and you're going to again use the ruler to draw the labeled line, the lines, right? The labeled lines and the lines that fall under your headings. So the title, you're going to use a ruler to underline that. You're going to need glue because you have to glue that picture, that drawing into your lab book, and of course you might need a scissors to do your little trimmings here and there. The geometry set is very important, as it would have some amount of drawing equipment and stencil that might very well help you to finish your lab nice and dandy, and the calculator to do your general calculations. Now, drawing versus diagram. I want to make it simple, and I really don't want to look at the content on the desktop. So if I'm talking and I'm not using what is on the desktop, forgive me, but what's on the desktop is correct. Your drawing is usually an accurate representation of the specimen. The diagram, on the other hand, it is a representation but not necessarily accurate. So the drawing might have a scale or magnification. Usually the diagram does not have this sort of a thing. Now, to calculate your magnification, it is what I love to call magnification equal drawing upon reel. So whatever you drew divided by the specimen, and we're talking about length here. So the length of a thing, given that the thing is proportionately drawn, the length of a drawing divided by the length of the specimen, keeping in mind the proportionality. And your answer must be written as X, whatever number you got when you did that division. The X must precede the number. Alright? A diagram, I said before, does not require a magnification, and we usually have a diagram coming right after our procedure, but of course before we get into the observation. Now, rules for drawing. The first rule, your drawing must be clear, accurate, and of course must be a true representation of that which is being drawn. Drawing must be done with a pencil. Your drawing must be done with a pencil. Now, if drawing will be part of what you are writing up, it's part of your observation, then the drawing should come before the content that you are going to write. Now, no color pencil nor coloring of a drawing, no coloring of a drawing should be done. You should not be using any color pencil for drawing unless you're doing like a diagram that probably shows a flow. It's not a drawing but a diagram. So if you're looking at the circulatory system, probably you want to put a little color in that if it is a diagram. But if it's going to be marked as a drawing, you should not use any form of color or coloring. Now, label line must be horizontal to the top of the page, and we're going to look at that when we get to the end of this presentation. If it is horizontal to the top of the page, when you extend the line, at no point will it meet the top or bottom of the page. Alright, so it must be horizontal to the top of the page. Now, labeled line must be drawn with a ruler. Don't try to use your label and your free hand to draw a labeled line. And the labeled line must touch whatever you are labeling. There should be no dot or arrows at the end of the line. There should be no dot or arrows at the end of the line that you're using to label. Very important point. Now, I know that when we draw, we have the tendency to bring out our artistic skill, the shading and all of that, but we don't do that in biology. And the reason we don't shade in biology is because the concept is that nothing is really solid. Things are made of small particles. So because they're made of small particles, we tend to allow the drawing or ensure that the drawing have the same type of a representation. So you are not going to shade any of your drawing. You can use stipples, you can use cross hatch, or you can use streak in your drawing. Now, if you are looking at a picture map, you are going to be using symbols. Alright, I hope that we understand. That is on screen, the streaks, the stipple, and the cross hatch. Now, the line should be smooth and unbroken. To ensure that you have a smooth line, ensure that you are well comfortable when you sit to draw. And once you put that pencil down and you start moving it, do not take it up until you are finished. The lines should be smooth and unbroken. So we really don't want to see where the lines are connected. Ensure that it is smoothly merged, that we won't see that. Now, draw only a small number of the small parts of the specimen. So if you were supposed to be drawing a die-cut leaf that has net venation, you don't want to draw the net venation right across the leaf. You just want to draw a small portion of it to show that the leaf has net venation. Now, labeling should have annotation. Now, in the diagram, I have left something to show you that this is a label versus this is a label with annotation. So once you draw, your labeling must have annotation. The lines used to label must not cross each other. So we don't want to see any line looking like a giant X or a web or a maze. The lines should not cross each other, and if they are horizontal to the top of the page, then there is no way they are going to be crossing each other. For the greater part, most of the drawing that we are doing in CXC, we can do them without getting to a point where we have to create a web crossing lines. Now, all drawing must have a magnification, and a magnification is written at the end of a title, and it is preceded with a X. What this X says is that the specimen X is whatever number times greater than what you have drawn for the greater part, unless you are doing a minification. But what we are doing is magnification. So So if you were drawing a elephant, it would have been a minification, but if you are drawing a leaf and you are drawing it bigger, then it is magnification. Now, labeling should be in one size font. So you don't want to be labeling the petiole and you're starting with a capital letter and then you're ending with common letters. It should be one size font. It's either you're going to be using all caps to label or all common to label. For me, I prefer use all common to label and all caps for titles. Now, title must be written in uppercase. The title is a description of the drawing and the view of the drawing. So a description of the drawing, I am drawing the upper surface, that is the view, of a dicotyledonous leaf. That's a description of the leaf. And then at the end of this, now you are going to have x2, meaning that my drawing is 2 times the size of the original dicot leaf. Labeling must be done to the right side of your drawing. Your labeling must be done to the right side of the drawing. Now, let us look at this. This is a picture I have taken from a lab book, and this leaf is, I'm going to be breaking down. I want you to follow my mouse here. Really want you to follow my mouse. So the tip of a leaf here is called the apex. Just below the apex here, we have what is called a leaf margin, or you might say the edge of a leaf. Now, as it stands here, margin is only a label. The margin is only a label. Apex by itself is a label. But when we have the little things that talk about the area that is been labeled, then that becomes our annotation. So let's look here. Net venation here is the label, and what we have written about net venation is the annotation. So we're saying net venation serves the transportation needs of the leaf. So these net venation, what they do is to ensure that substances are taken to the various parts of the leaf and are removed from the various parts of the leaf. Then we have mid-red, we have lamina, and you can look at the diagram as is. Now try as much as possible to get these lines even straight down. Realize, try to get it even. And when we say it must be parallel to the or horizontal to the top of the page, it means that it must come the same width between here and here. When you get to the end here, the width must be the same. So it is horizontal to the top of the page. All right, we spoke about not drawing too much of the small details of the specimen. Now this is a cardinal leaf, and you will see it later. If you observe here, we have put in the net venation, but we have not done the net for the entire leaf. We just took out a little part, because doing that can sometimes get the diagram messy, and you know, we really don't want to go there. All right, so this here, we're supposed to be looking at the title, and it should tell the view and should be a description of what we drew. So if you look at what we have here, it says that it's a drawing of the under side, that's the lower side of a caronilla leaf, and it is saying x1. What that means is that the drawing is the same size as the original. Now this is the Carolina plant. If you are in Jamaica, it is called rice and peas. Locally it is called bees bush. It has medicinal properties. I think it is used to really deal with things like, of course, the underside, right? You are looking here at the upper side, all right, or the top of the leaf. Right, we went through this just now, so we are pretty much at the end of the presentation. There is a link to actual drawing in my description. I want you to ensure that you have subscribed to this channel, you have liked this channel, and you have shared this channel with some friend from some other school. please leave a comment so that we know how well we're doing and what we can modify and how we can help you. All right, it has been a pleasure serving you. Be safe, practice your drawing skill, and get ready to get your drawing in your lab book. One tip about lab books: remember, all pages in your lab books must be labeled. Second thing, remember, the date that the lab was done must be written in the lab book. Peace out. Walk good.